Right, let's get started. So welcome everyone to SADS Foundation's webinar on gene therapy. Appreciate you joining us tonight. I'm Marcia Baker, the Program Director for the SADS Foundation. We're joined again by Dr. Andrew Landstrom, who will provide an overview about gene therapy, what it is, and how it works. Dr. Landstrom, as you remember, has spoken to us before on this topic in previous webinars. He assures me he's updated his presentation to include some recent developments. We'll take questions following his presentation, and you can post those in the chat. Dr. Landstrom is an associate professor of pediatrics at Duke University of Medicine, School of Medicine. He's a physician scientist, so an MD, PhD, cross-trained in pediatric cardiology, electrophysiology, human genetics, and molecular physiology. He directs an active research program at Duke University School of Medicine that brings together clinical medicine, functional genomics, precision medicine, and molecular and cellular biology. Dr. Landstrom is also a member of our SADS Foundation Board of Trustees. We are honored tonight that he's volunteering his expertise and time so generously for the SADS Foundation. So welcome, Dr. Landstrom. I'll turn the mic over to you.
Thank you, Marcia, and good evening, everybody. I'll try to share my screen, and hopefully this works the first time. How's that look?
Perfect.
All right, well, good evening again, everybody. It's always a pleasure to get the chance to talk to all of you. Thank you for sharing part of your evening with me. And I have about 40 minutes or so slides worth of slides for us tonight. And then we'll have plenty of time towards the end to answer any questions that you may have. But I'm really excited today to talk to you a little bit more about Gene Therapy 101. What do I need to know about gene therapy for SADS conditions? So importantly, I have no disclosures or conflicts of interest or relationships with any of the for-profit organizations that I'll mention today, which I hope you find gives you an unbiased view of gene therapy. I do receive research and salary support from a number of not-for-profit and federal organizations, though. So in our time together, I hope to cover a few questions that you might have front of mind. We'll start with the basics. What is gene therapy? We'll give a brief overview of the history of gene therapy and some missteps that happened that I think have directly impacted how we think about gene therapy today. We'll talk about recent advances. Why is it that gene therapy now is coming into the forefront of clinical medicine? What is CRISPR? You may have heard about this in the public media. We'll be able to talk a bit more about it. And what is AAV, which is a kind of a common thing you may hear when you talk about cardiovascular gene therapy. We'll talk about how cells are targeted and how genes are edited or delivered into cells. We'll talk about active trials, particularly for SADS conditions. We'll talk about how one can stay more active and learn more about clinical trials that are on the horizon. And then we'll take a step back and talk about gene therapy more broadly in our last few slides. So really simply, gene therapy is any genetic modification to a cell that produces a therapeutic effect. And I think oftentimes when we think about gene therapy, we think about changing the DNA, and that can be a gene therapy. But oftentimes, as I hope to show you, it's actually sometimes a little bit more than that. We're actually trying to introduce things, genetic therapies directly into cells to make up for a genetic defect. And so really any discussion about gene therapy needs to kind of center around a common language. And that language is around how changes in DNA can cause disease. So if we think back to high school biology, the central dogma of molecular biology is that we are who we are because of the proteins that we make in our cells. And all of those proteins come from DNA. DNA is like the cookbook. It's like a recipe for making all the proteins. And that DNA makes RNA. That RNA then passes that message to ribosomes, which make the protein. And that protein folds and goes somewhere in all of our cells. And that's what makes us who we are. And so when we think about genetic disease, like cardiovascular genetic disease, I tend to think of it in two big buckets. The first bucket is a misspelling in the DNA. We call these a single nucleotide change or a single nucleotide variant that changes a letter in the gene that then gets passed through the RNA to make a change in the protein, and that can disrupt how that protein actually works. Conversely, there's another kind of misspelling in the DNA, which is a little bit more than a single nucleotide change, more than one letter. It's actually a bigger change. We sometimes call them loss of function changes that actually disrupts the entire gene, the entire message, and the entire protein. And oftentimes, these loss of function mutations cause loss of the actual protein. So you can imagine in the column to the left, if you have a single misspelling, that mutant protein is being expressed. In the one to the right, there is no protein. And so right there, depending on the kind of mutation one may have, the approach to correcting that might be very, very different. Okay. And so, the basic premise behind gene therapy is, well, if there's a defect, we should fix it, right? And so if you think about a heart cell, that heart cell has a gene. That gene makes, say, an ion channel. That ion channel is needed for the proper functioning of the heart. And this is the case for many SADS conditions. There's a mutation in that gene, a mutation in the ion channel that disrupts the electricity. So if you have a loss of function mutation, you may have fewer of those ion channels being made. If you have one of those missense variants, those single nucleotide variants, you may have abnormal ion channels made next to normal ion channels. Both can give you arrhythmias, but both will have very unique ways that they could be treated. For example, if you have a loss of function mutation, you may just want to give back the correct gene. That may not work as well if you have that missense variant, that single nucleotide variant, because simply giving more gene doesn't do anything to correct the abnormal protein. So kind of right there, a blueprint for maybe how understanding one's genetics is really central to understanding how one might treat that disease. So like in all things, there are many, many problems that might occur. And one of them is delivery. And so for the longest time, delivery, how do you get medicines into the cell? How do you get gene therapies into the cell was a really big question. And this is a question that actually has a lot to do with the cell itself. So if you think about the heart is made up of heart cells. That heart cell has a little membrane around it, which keeps everything within that cell. If it's a heart cell, we call that a sarcolemma. It's also called a plasma membrane. It's really just sort of like a fatty layer that goes around the cell, but it's actually much more than that. It's actually used to protect the cell from invaders, specifically against viruses and things. And so even though it is just a little oily layer, it's really a really impenetrable barrier. And so getting things into that cell can be really quite challenging. One of the best ways that we know about is actually a virus. So viruses make their business by injecting their contents, usually it's DNA or RNA, into a cell. And so what if you took out all the bad things out of that virus and loaded it with things that you wanted the gene or you wanted the virus to deliver, say, for example, a missing gene? One could then use a virus and deliver that gene product. And the basic premise is if you have a defective gene A that's not being made, you could load up a virus with that normal gene, infect those cells with that virus, which then will express the normal gene. And this has been sort of the approach for these loss of function mutations for many, many years. And in fact, this was the first gene therapy approach that was tried. So way back in 1989, the first patient was successfully treated with gene therapy. And this is her, Ashanti De Silva. Ashanti had not a heart condition, but she had a very severe immune condition called severe combined immunodeficiency, which, because of one of those loss of function mutations in a gene that one needs to make a protein that's really important for the immune system, she would have had a very disrupted and abnormal immune system because of that mutation. And so she got a gene therapy to replace that missing gene. And it worked. And she actually went from having almost a non-existent immune system to actually being healthy. And in fact, she's alive today, and she's a genetic counselor of all professions. And many of you might have intersected genetic counselors. They're part of our cardiovascular genetics medical community as people who are really important for helping to define and care for genetics patients. And so she's one of these genetic counselors and a real success story for gene therapy. Um, but just a few years after that, there was the first death. So 1999, a name that many of you may have heard of if you've heard about gene therapy deaths, Jesse Gelsinger. So Jesse had this really rare disease called ornithine transcarbamylase deficiency, which basically meant he had a loss of function mutation and couldn't make some of the chemicals that his body needs to survive. And so the thought was, well, maybe we can give this gene therapy to him. And Jesse died. And in retrospect, it raised some very significant questions about maybe Jesse was actually too sick to get the gene therapy. Uh, did the gene therapy researchers who were trialing this new therapy properly consent him and his family about the possible side effects, including death for gene therapy? Um, and then there were other patients who had some very severe side effects as well. And so because of this, there were a lot of challenges that were raised back in the 1990s to the early 2000s about, how we're going to have to get over these challenges if gene therapy is ever going to reach prime time. And these challenges were many. We talked a little bit about sort of, getting viruses into cells. These deaths really highlighted the need to address other problems with gene therapy. A big one were off-target effects. That is, if you have a piece of DNA that's being inserted into a cell and maybe even inserted into the native DNA, we want to go to one place and one place only and not go somewhere else. That can cause cancer, for example, if it goes into a wrong area and turns on a gene that shouldn't be on all the time. Gene therapies at the time were not very tissue specific, meaning you might have wanted it to go to the heart, but say it went to the liver, it could cause liver inflammation and liver failure. That's a big problem. The immune system, our native immune system, is designed to identify viruses and to kill them with an immune response. And that immune system reaction can be good normally when we have viruses, but can lead to these really bad side effects that people can have. And back then, because of the limited technology, gene replacement was really the only strategy. So you could only put in a native gene. You couldn't do anything else. And really, these challenges really kind of pressed pause on gene therapy for nearly 2 decades. But science continued. And there have been a number of, I would say, breakthroughs, really, that have accelerated our ability to get over many of these barriers. And I'll share what those breakthroughs are. This is a big one, the human genome. So back in 2001, it cost roughly $100 million to sequence a human genome, one human genome. On the y-axis here is the cost of sequencing a genome on a log 10 scale, and on the x-axis is time. As you can see, it has fallen faster than Moore's law, which is that every 18 months the transistor, speed increases, doubles. It fell faster than Moore's law would say something should increase, right? So a precipitous fall in the cost of sequencing a human genome. In fact, at Duke, we can sequence a genome now for $400, which is really quite inexpensive. So that's been a major accelerant. What this is fueled is people can get gene testing now for a lot cheaper. More people can be identified who have genetic cardiovascular diseases, for example. Another major breakthrough is CRISPR-Cas9. You may have heard of this in sort of the public media, but CRISPR-Cas9 was actually first found inside bacteria as a defense for those bacteria against viruses. And what it is, it's like a little immune system inside a bacteria. It's a set of molecules that recognize very specific parts of DNA or RNA and chops it up. And really, if you start to understand that, you can start to use CRISPR-Cas9 as a very precise pair of molecular scissors that can go in and target DNA and RNA, make precise cuts, and then you can put in other pieces of DNA that you might want to replace that with. Because when the cell is fixing a cut, it looks around for a template, a recipe for how to fill it in. And if you provide it with something a little bit different, you can actually change the DNA in a very precise way. Further, CRISPR-Cas9 has provided a lot of molecular tools to manipulate DNA, and this has really accelerated the ability to make genetic tools. The models for studying cardiovascular disease have gotten better. So these are beating cardiac myocytes, but they're not from a heart. they're actually derived from somebody's blood. So these are called induced pluripotent stem cell derived cardiac myocytes, which is basically a long name, meaning we can take a little bit of blood, we can isolate a certain kind of immune cell from your blood, and then trick those immune cells into becoming your stem cells, and then guide those stem cells into becoming heart cells, and they beat in a dish just like this. And what this has done is it's allowed us then to study human heart cells with various diseases in a dish that wouldn't have been possible otherwise. So a very important breakthrough for us to model disease. And then finally, the virus, our ability to make viruses has gotten a lot better. So we've identified particular viruses that have a predisposition to go to the heart. This is one of them. This is AAV9, which is an adeno-associated virus 9 that really has a predisposition to want to go to the heart and is really an optimal one for gene therapy. And so really, there's improved delivery. You can give gene therapy any number of ways. You can give it just by placing DNA or RNA or protein onto cells. You can use microsomes, these little bubbles of lipid that can transfer things. You can use nanoparticles, and you can use viruses. But really, adeno-associated virus, AAV9, is really great for cardiac gene therapy. It has better tissue targeting. It wants to go to the heart. It doesn't have quite that immune response that some viruses do. And then the cargo, whatever it delivers, can be made forever in the cell, in theory. And so really, because of these advances, I think that gene therapy holds tremendous promise for SADS conditions. So let's tackle a few SADS conditions, and we'll talk about sort of where the preclinical science is, that is the science in models that may get us to trials, and then if there are trials, where those trials are. So first, let's talk about long QT syndrome, which many of you may have or may have heard of, which is basically defined by a long QT interval on an ECG. And that just measures the amount of time it takes for a heart to reset electrically after it beats. And if you have a long QT, it means your heart takes a little bit longer, a few milliseconds, sometimes longer than normal, to reset than normal. And that can make the heart a little bit unstable electrically. Many of the different types of long QT syndrome each have their own specific ways they cause disease based on the particular gene mutation or the gene that's being changed. So for example, long QT syndrome type 1, which has mutations in the gene KCNQ1, it's one of the potassium channels of the heart, is well known because it makes you get disease from this type of long QT syndrome because of misspellings in one copy of the 2 copies that we have of this gene. And because of that, there's normal gene that's being expressed, and there's mutated gene that's being expressed as protein, roughly in the 50-50 kind of a blend, we'll say. So you can see a situation where you really can't, say, deliver normal gene because you've still got that mutant gene in the cell. And so this is work actually done by Mike Ackerman, who's the president of SADS, out of his research group, which has shown that you can actually silence the expression of the bad protein and recover the normal expression of the wild type protein, something that he's dubbed suppression and replacement. And this is just some of the data from the initial paper. So you're actually seeing the science in the public domain. These top panels are tracings of heart cells, the way that heart cells depolarize and repolarize. And the x-axis here is time. So you can see that some cells depolarize with this upward spike and then repolarize and come back down to baseline. You can see that some of these cells in this red line have a wider width to them. And that's because they take longer to reset than the other cells, which are normal. So in the black here are normal cells. In the red cells, those are the long ones. Those are long QT cells. And then you can see when you actually deliver this suppression replacement gene therapy, you get the blue line, which is actually very similar to the black line because you're normalizing the long QT of those cells back down to normal. All right, and they can actually quantify that here, looking at different measurements of how long it takes the heart to reset. He has also shown this for long QT syndrome type 2, which are due to mutations in the gene KCNH2, using a very similar suppress and replace strategy. So again, these are those cells. The wide cells, the ones in red, are the ones that have long QT. The ones in black are the ones that are normal, and the ones in blue are the ones that have long QT but got the suprep gene therapy. And as you can see in these situations, it is normalized back down to baseline, which is really quite exciting. What about for Brugada syndrome? Well, Brugada syndrome is due to mutations in the sodium channel, SCN5A. But Brugada is pretty challenging because the sodium channel is really, really, really big. Too big to fit into most adenoviruses. And so here's a really interesting paper from 2022 that actually kind of did a molecular trick. So there is this protein called MOG1, which kind of has a funny name, but actually what it does is it wants to grab a hold of the sodium channel when it's inside the cell and rush it to the membrane. It's called a trafficker or an escort. It wants to bring that normal protein up to the membrane. Patients who have Brugada syndrome have a mutation that's loss of function. So they don't actually make enough of that sodium channel. So their idea was actually very clever. What if we actually just put in this chaperone, this escort for NAV 1.5, the sodium channel? Would it actually grab more of the normal sodium channel and bring it to the membrane and fix the mice? And they actually used as their experimental model mice with a mutation that had Brugada syndrome. And this is the data from that paper. They actually found that when they gave that MOG1, that escort, that chaperone, it actually normalized the loss of the sodium channel that was experienced in those mice, which I think is really, really cool. They couldn't replace SCN5A. It's too big, so they went with a little escort that actually encourages the normal NAV 1.5 to get to the membrane. So that's really, really cool. Again, this is all preclinical, not in clinical trials. This is done in mice, but I think a really cool proof of concept that you can do something like this. What about CPVT, catecholaminergic polymorphic ventricular tachycardia? So CPVT is due to mutations oftentimes in a gene called RYR2, sometimes in other genes like calsequestrin. But what ties all of CPVT together is that they tend to get arrhythmias with exercise or with high emotional states. The C in CPVT is for catecholamine, adrenaline-fueled arrhythmias. And there are mice that actually have CPVT. This is a mouse with a mutation in RYR2. And if you take this mouse and if you run them, or if you give this mouse adrenaline, they will actually get the same ventricular arrhythmias that patients with CPVT get. RYR, and this is a particularly challenging mutation too, because CPVT mutations are not loss of function. So they're actually point mutations that tend to cause RYR2 to be a bit more leaky, to gain function. So how do you actually fix that? RYR2 is far too big to put into a virus. And how would you fix a leaking mutation anyway? And so they actually came up with a little small molecule that actually inhibits a protein that can irritate RYR2. That protein is calmodulin kinase 2. And they put that little tiny inhibitor into AAV and gave it to the RYR2 mice. And so what you're looking at here, these are actually heart cells to the left that are being paced. Those flashes, those upward strokes are beats of each of those heart cells. And they actually gave these to mice. And as you can see, the mice who had CPVT had all kinds of arrhythmias. See how kind of all those extra beats there are there, the really irregular beats that are not found in normal mice. But if you give them this inhibitor of calmodulin kinase, which is an irritator of RYR2, all of it went away. And they actually gave it to the mice, and their arrhythmias went away too. So really, really cool. Again, done in mice, done in cells, not done in people, but interesting preclinical work that you can actually use inhibitors of upstream modulators to actually silence a downstream protein, which is really, really cool. And then finally, arrhythmic cardiomyopathy, which is defined as fibrofatty replacement of the normal heart muscle that can cause arrhythmias and sometimes heart failure. What about this? Well, the biggest gene that's mutated in arrhythmic cardiomyopathy is a gene called PKP2, which makes a protein called plakophilin 2. And these are classic loss of function mutations. So patients who have arrhythmic cardiomyopathy because of PKP2 mutations, they generally have 2 copies of the gene, and one of them doesn't work because there's a mutation in that gene. And so this is, that sort of more straightforward, traditional like gene replacement strategy. And Tenaya Therapeutics, this is from the public domains. This is research that they've published at one of our gene therapy meetings. Said, well, why don't we just actually replace this missing PKP2 in a mouse model of PKP2 arrhythmic cardiomyopathy? And this is that work from Tenaya that's all, again, in the public domain. And what they basically found was they took these mice with a PKP2 mutation. They found there was arrhythmic cardiomyopathy in those mice. And then when they gave the PKP2 back using the virus, those mice actually got a lot better. It prevented them from getting sick. And then if they were sick already, it actually recovered some of the squeeze of the heart, some of the mortality of these mice, and actually made many of the arrhythmias go away. And this has really fueled, this preclinical work has actually fueled the first trial in a SADS condition, which is now being started as a phase 1 clinical trial by Rocket Pharma for a PKP2 arrhythmic cardiomyopathy trial. And so they are right now enrolling individuals in UC San Diego and CHOP for this phase 1 clinical trial, looking for 18 years or older folks with PKP2 mutations who have severe enough disease to require an ICD, but a preclinical model being translated now to a phase 1 clinical trial. Now, despite this excitement and despite the success of these preclinical models, it's important to realize that there are challenges still that are facing gene therapy. This is one that you may have heard of. It kind of made a big splash in our field as sort of a trial that failed. This is the CUPID-2 trial. And the basic premise here was that sometimes people with heart failure, and this is not a SADS condition, these are people who maybe have heart attacks, they have ischemic heart disease and develop heart failure, the heart doesn't squeeze very well. We know that one of the proteins that's lost in that heart failure is a protein called SERCA2. SERCA2, the idea was if you could give it back, you may actually encourage the heart to beat and get through some of this heart failure. And all the preclinical models look great. They actually went all the way to a phase 2b clinical trial, and they gave adenovirus with SERCA2 into the coronary arteries of people who had heart attacks and heart failure. And essentially what they found was it didn't seem to matter. So the individuals who got the placebo and the individuals who got the SERCA2 had the same number of cardiac events, had the same survival. It didn't seem to have any benefit. And we really don't know why. There's some thought that maybe the virus wasn't delivering its payload, but you can't take pieces of people's heart and look at them. These people are all still alive. So it really, we don't know why this failed, but it did. And importantly, there actually have still continued to be some deaths. And it's important to realize that. And so this is a rather I guess, sort of well-known case from 2021. Astellas or Adentis had a clinical trial for using AAV to treat a myopathy, a skeletal muscle disease. And they gave it to a number of individuals to test safety. And over a year after the virus went in, they had 4 deaths. And it was really a tragedy. Why is it that these individuals who seemed to do well for a year then had these deaths very, very late? And this is somebody you may have heard about. This is Terry Hogan, who has Duchenne's muscular dystrophy, which is due to mutations in a gene called DMD. And he rather famously became what we call an N-of-1 clinical trial, where they were going to deliver CRISPR-Cas9 to fix Terry's gene mutation. And he died. And again, this happened about a year ago, unclear why he died, but lots of concerns about, potentially the virus or how it was delivered. But whereas, 20 years ago, the field pressed pause, right now, there does not seem to be any slowing down of the gene therapy pipeline. So what does the landscape look like for gene therapy overall? And so this is the genetic landscape, the gene therapy landscape as of the end of 2023. So you're really up to date now. There are 30 gene therapies that have been approved by the FDA. There are 29 RNA therapies that have been approved. There's 65 cell therapies that have been approved. Nearly all of these are for cancer, skeletal myopathies, metabolic diseases, not the heart. There are just under 4,000 gene, cell, and RNA therapies that are currently being in a sort of pipeline, we call kind of a preclinical pipeline of discovery through FDA registration. And there are just over 1,000 therapies for rare diseases in the sort of preclinical through FDA registration pipeline, but nearly all are cancer related. This continues to be a big business too. And so there's a lot of money actually going into companies that have ideas for gene therapy. You can see it's waxing and waning a little bit over time, but in last fiscal quarter, Q4 of 2023, there was what, $600 million, actually almost $700 million raised for sort of financing of these gene therapy trials. Again, nearly all of them are non-cardiac or non-SADS conditions, but I think that's going to change. So how can you stay informed in a rapidly changing field? Well, the biggest piece of advice I can give you is go to trusted resources. Here's a good one, clinicaltrials.gov. It's a little bit cumbersome, as I think many kind of government websites are, but you can go into the website, you can enter into the fields a condition and other search terms and ask, what are the ongoing trials in this space? And so, for example, if you entered ACM, arrhythmic cardiomyopathy, you would see that phase 1 clinical trial by Rocket Pharma, PKP2 ACM, the one that we talked about, where they're probably replacing PKP2. So you can go here, clinicaltrials.gov, and you can search for your disease and see all the trials, not just gene therapy, but all the trials currently being conducted for your disease, and trials that have happened in the past too. Another trusted voice is SADS. The SADS Foundation does a lot of education in this space. You're at one of the webinars right now. There are a lot of materials that SADS puts out so that you all know about these really cutting edge discoveries as they're happening, and it's a very balanced perspective. I'll also draw your attention to an upcoming, what's called an externally led patient-focused drug development meeting, ELPFDD. And essentially, this is a way for you all to have your voices heard, which are often, almost drowned out in the kind of, crush of science and ongoing trials. But it's really tremendously important that you all, patients and your lived experiences, inform how we and other entities like the FDA view the march of gene therapy for rare diseases. And so if it's something that you're interested in, SADS is going to be doing an ELPFDD for long QT syndrome and catecholaminergic polymorphic ventricular tachycardia. You can learn more about it. And if you're so inclined, you can support the effort financially. As you can imagine, SADS is a not-for-profit organization. This is central to their mission. It can be a little bit financially challenging to do these things. They do it because they feel it's central to mission, and you can support them financially if you're inclined. And so really, it's been a long road, but I think, there's been some really amazing discoveries. I think the field is still really exciting for cardiovascular gene therapy, particularly for SADS conditions. We have a long way to go ahead, just to make sure that the therapies that are being delivered are safe, that they're going to be efficacious, and that they're going to be able to take away some of the morbidity, mortality associated with these SADS conditions. And so my take-home points are improve AAV transduction efficiency, cargo delivery, CRISPR gene editing, among other advances, have driven a resurgence in gene therapy for a variety of illnesses. There's some exciting preclinical data that's shown that gene therapy can be useful for things like long QT, short QT, CPVT, ACM, and other SADS conditions. At the end of the day, careful clinical trial work and more science is needed to realize the full clinical benefit of gene therapy, and I think we'll get there. And so with that, I thank you for your attention, and I welcome any questions. Thank you very much.
I guess I'll go first. This is Jorge McCormick. I'm a pediatric electrophysiologist and been member of SADS for a long, long time. And I'm just having flashbacks of 20, 25 years ago when Mike and I were I was sending him Mike, long QT cases, and he was doing the genotypes, and we were being able to do cascade testing with those who were positive. So this was a phenomenal lecture, and I congratulate you for that. Let me ask you a question though. So in the case of a arrhythmic cardiomyopathy, unlike long QT syndrome, in long QT syndrome you have viable cells that are irritable. In arrhythmic cardiomyopathy, you have premature apoptosis, you have cell death, and that cell death becomes a substrate for arrhythmias. So what is the expectation of gene therapy for arrhythmic cardiomyopathy if you already have cell death? You're not going to resuscitate those cells. How is that going to work?
I love the question. Thank you. So I think, there is sort of what we would expect based on the preclinical work, the mouse model work. And then there's humans, and humans sometimes don't read the book when it comes to the preclinical model.
Right.
With that grain of salt, I will say that at the end of last year, the PKP2 knockout mouse, the one that gets signs of arrhythmic cardiomyopathy, and it's full-blown fibrofatty replacement, the fibrosis of the ventricular myocardium, loss of function, terrible arrhythmias, and in the knockout mouse, when you knock it down conditionally, as within weeks of losing PKP2, the mice, their hearts fail and the mice die. The Tenaya therapeutic trial, and then this paper published in Science, I believe, or maybe Nature Cardiovascular Research, 2 months ago, it found that if you actually let those hearts remodel and get the mice just before mortality ensues, you actually recover some of the functional myocardium, which is amazing to me. You actually suppress the arrhythmias, and you actually get back some of the contractility that's lost, at least by transthoracic echocardiogram. So at least some preclinical in vivo work that actually that fibrosis may not be as set as we once thought it was. Now, I'll be the first person to tell you that people are not mice, and many things that look great in a preclinical model simply don't translate. But that was a pretty surprising finding. I will say that I think there is a lot of interest in that sort of fibrous remodeling aspect outside of SADS conditions, with some really interesting therapies that go beyond just gene therapy too. We didn't talk about it, but there's cell therapies. Can you actually target, say CAR T therapy, right? Engineered T cells that go in and destroy, say, fibroblast cells within the myocardium. Very interesting work there too. So I think there's a lot there. Now, whether any of it has shown in clinical trials, that's still an open question. But I think in the preclinical space, there's a lot of optimism.
Excellent. Thank you.
Of course.
Thank you, Dr. Landstrom, for that wonderful presentation. And thank you, Dr. McCormick, for starting us off with the Q&A session. If anyone else has questions, you can post them in the chat, or if you want to ask it verbally, just raise your hand. I do have one already that was sent to me. Andrew, it's who will be eligible for gene therapy clinical trials?
I think ultimately it's going to be up to the trialists. The people who are eligible now. So if you think like a company, right? So if you're investing millions and millions of dollars into a gene therapy, you want to be able to show that gene therapy is safe, that gene therapy works. And so they are under sort of a financial incentive to pick individuals who they think are going to respond well. Those individuals tend to be a bit more later stage, more advanced in their disease. And they tend to be sort of carefully selected because the last thing they want is somebody who's too sick to get the gene therapy. Think of Jesse Gelsinger, who died and potentially was too sick to get the gene therapy. But they want somebody who, there's the ability to see a benefit from. And they call it sort of this Goldilocks phenomenon of somebody who's not too sick, but sick enough. I think where at all possible, individuals will probably be adults, just because if you're trying to think about a pediatric, a vulnerable population for gene therapy, not impossible. There's gene therapy clinical trials right now, but that, being a vulnerable population does kind of layer another layer of complexity onto it. If you look at who's being enrolled in the phase 1 clinical trials, they tend to be individuals who are adults, 18 years or older. They tend to be individuals who are a little bit more sick. So advanced disease, where maybe the medications that are generally felt to be frontline are not as effective. Those tend to be the individuals, adult and a bit more sick. Now, those first trials are the trials to show safety and then to show efficacy. And then inevitably, over time, my hope would be that the gene therapy would be given to more and more people, provided it's safe and efficacious, and not just a very targeted population, but the trialists are under a financial incentive to kind of, show early success.
Thank you. Another question. What questions should I ask before joining a gene therapy clinical trial?
I love the question. Well, the first thing is, do you know your own gene mutation? So if you have a SADS condition, it's really important that you know your genetic mutation. If you haven't been gene tested, I would say get it done. It should be standard for everybody with a SADS condition. You should know your gene mutation. Second question is, make sure that you're having this conversation with somebody who feels comfortable talking to you about where the state of the art is in cardiovascular gene therapy. And that may be a specialist. And so if you feel like you're not getting answers from the person in front of you, you can actually seek out SADS conditions experts. And SADS can help you find those experts near you who can have that conversation with you. Then I would be asking very specific questions. If it was me or if it was my family, I'd want to know, one, is there even a gene therapy trial out there that's in phase 1 or phase 2? For a lot of the SADS conditions, those trials do not exist yet. My hope is that in the next few years there will be, but for many SADS conditions, there's not a gene therapy trial. If there is one, what stage is it? If it's stage 1, it may be that they're just looking for safety. And, these are big commitments. So for example, you may have to live in a town where they're doing the enrollment for that stage 1 trial for a little while, because that person may be admitted, lots of testing, et cetera, et cetera. So it's a big thing to take part in a clinical trial, particularly in those early stages. So the next question is that even conceivable for me and my family? And if so, who would I go to? A lot of these trials have patient advocates who can provide you with more details if you would like. And also that SADS disease expert can also provide you with some details as well too. But find those trusted sources, get more information, and know whether you might be eligible based on your genetics.
Excellent, off the cuff. Very good. Now, I have a question that Christy's posted to everyone. It's a hard question, she says, to answer, but in general, in the medical community, how do cardiologists overall see gene therapies? Yeah, i.e., is it going to be difficult to get a patient's EP or heart failure specialist to get on board with their patient's participation? Comments?
I would say the community is varied. I think if you are being seen by an expert in a particular SADS disease, I think they're probably more comfortable talking to you about gene therapy, probably a little more comfortable knowing what the gene therapy options are. I think if you're talking with somebody who's a bit more, maybe a general cardiologist, more of a general practitioner, they may not feel as comfortable. So I think that's sort of one sort of way of looking at this. The specialists themselves, we talked about kind of important things to ask, is does that specialist have a relationship with the trial in any way? Because that would potentially be something that's important for you to know. If that individual recruits for the trial, if they have a scientific interest in the trial, that's an important sort of disclosure that they should have with you. I think overall, if you have questions, your physician is the great first step. That SADS expert is a great second step. And then there's lots of information out there publicly. And organizations like SADS can help provide that information as well too. So I hope no one ever feels like there's nowhere to go if they feel like they can't get the answers from the medical professional in front of them. There's lots of resources if you do a little digging, but again, go for those trusted voices.
Okay, thank you, Andrew. This one was sent to me. What are the suspected reasons for the more recent deaths that occurred a year after receiving gene therapy? Do you
And
know?
these are real tragedies. I mean, my goodness, can you imagine having a therapy and a year later being faced with these tragedies? It's not really clear. Again, I'm looking at it from what's available in the public domain. I have no insider knowledge at all. It's possible that many of these are sort of late reactions of the immune system, particularly in certain organs like the liver. But again, it's really not clear. For Terry Hogan, still not clear how he died, which, I think is in some ways, not as satisfying as we'd want.
Okay. Scott posted, how long does a clinical trial take to complete? His daughter has the PKP2 gene for ACM.
Probably not as fast, Scott, as you or I might want, to be perfectly honest. Phase 1 clinical trials can take years. They're trying to find a handful of patients to look for safety, and then they follow them for a long period of time. And so it only goes as fast as that last person recruited going through the trial. And that's phase 1. There's phase 2. They can split that into multiple subphases, phase 3, et cetera, et cetera. It can take, on average, 10 to 15 years. Again, times vary, but that's sort of the ballpark that I would have in mind. And I would love for these trials to go on a faster timetable than that. But they're designed to be slow for a reason, because safety is paramount. The ability to show clear safety, and that if there are potential side effects, that they are dramatically outweighed by the benefit of the therapy is really important. And so that's the timeline, and it's patient safety that really drives it.
Thank you very much. Are there any other questions? Seeing none, I wanted to thank Dr. Landstrom very much, and also give a reminder for a webinar that we are planning now for April. And just watch our emails for the details, but it's going to be about clinical trials specifically for PKP2 ACM. And we'll have an overview by an adult EP and companies that have approved clinical trials for PKP2 will also be available. Some questions, and we'll have some breakouts so people can ask specific questions of each company about their particular clinical trial. So watch for those details. So we appreciate your participation tonight. Thank you very much for your questions and engagement. And thank you very much, Dr. Landstrom.
My pleasure.
Good night, everyone.
Good night.